variability and the association character for yield and yield related trait on soybean genotypes. In Ethiopia and western part of Oromia. When I came to outline of my presentation, introduction, objectives, materials and the methods, result and the discussion, conclusion and the recommendation, and lastly, acknowledgement. When I came to under introduction part, soybean is the most important legume crops which is used for human food consumption, for feed of animals, and for agro-industries, and for different factors for nutritional purpose. It is considered as a king of legume crops, which is used for high protein and oil content, which is 40 and 20% respectively. Soybean is the most important pulse crops, which is essential for amino acid composition, for rather than cereal crops, it is high protein content. When I came to this agroecology, soybean is the most important, which is growing midland and up to highland with a rainfall of 500 up to 700 milliliter and with annual temperature of 20 up to 25 degrees Celsius across the world. When we conduct this research, according to FAO organization, which is estimated around 320.5 million metric ton, which is the major production in the world is Brazil, USA, and Argentina. And from Africa, South Africa, Nigeria, and Zambia is the most consequential producer in Africa. When I came to around our concerning to our description of the title, Ethiopia is the fifth producer from Africa, which is rank soybean and pulse oils with oil crops, which is the most important. National average in Ethiopia, which is around 2.27, according to Statistical Agency of 2017. Still in a research area, it is limitation. When I came to statement of problem or gap of this research, narrow genetic base and low productivity potential of this soybean crops, which is around our experiment, which is contaminated by soil fertility, weak value chain and the market linkage. Also foliar disease, which is as a statement of problem. So for this purpose, genetic variability and studying on diversity of this crop is the most important around the study area. Quantifying the genetic variability and the diversity is the most important. When I came to under the producer, soybean germplasm is focused on adaptation and evaluation around the Wollega zones. Because of this, we selected this title to study under research area, which is the most important for increment of this soybean production. Objective of this research is to estimate phenotypic coefficient variation and the genotypic coefficient variation and the genetic gain of soybean genotypes. Now you are hearing. Hello, everybody. Hello?
Yes, we can hear you.
Okay. And the second objective is to estimate the magnitude correlation among grain yield and the yield related traits. And the last objective is to cluster soybean genotypes genetically across Ethiopian agroecology for production of this, the most important soybean crops. Material and the methods. The experiment was conducted at the Wollega University research site, a western part of Ethiopia, which is far from Finfinne, around 357 kilometers and 1,319 meters above sea level. And the ecology of this average of rainfall is almost 1,200 millimeters, which is suitable or possible to grow for the agroecology of soybean crops around the western part of Oromia. Maximum and the minimum temperature was 32 and 60 averagely. Experimental material, we use for the experiment 97 accessions of soybean industry released the varieties, which is Niala, Afghat, and the Karlak. This experiment was laid out lattice design, simple lattice design by 10 by 10 simple lattice design. Because of this is a large, when it is a large genotypes, we use these designs in order to manage homogeneity of the soil fertility and the ingredient of the slopes. Spacing is as a recommended 60 centimeter between rows and 5 centimeter between plants. It is for recommended spacing for soybean genotypes. This experiment was conducted July 2080. All agronomic management practice, including fertilizer, from sowing to harvesting, according to the recommendation of this crop, we was conducted. When I came to the under data collection, the following parameter was collected: days 250 emergency, days 250 flowering, days to 95% of maturity, and the grain filling period were recorded. Under vegetative data, plant height was measured in centimeter, and the number of primary branches and the number of nodules was collected. Under yield and the yield related traits, number of pod per plant, number of seed per pod, under seed weight, biological yield, grain yield, and harvest index were recorded. When I came to under data analysis, data was analyzed by Statistical Agency of SAS software version, and the efficiency of simple lattice design was compared with RCBD, which is for our analysis of data. Mean difference was tested using student Q&A test at 0.0 alpha levels. When I came to under the estimation of genetic parameters, which is in order to use for measurement of genotypic and phenotypic coefficient variancy, which is phenotypic variancy, which is equals to variance of genotype plus variance of error over replication. Phenotypic and the genotype coefficient variation for each trait was computed by this using this formula, which is equals to phenotypic variance over the grand mean of each trait times 100. When I came to under heritability is the most important factors for metric trait in order to measure the metric trait for selection, for direct selection and indirect selection for future hybridization of these genotypes, which is equals to genotypic variance over phenotypic variance times 100. Under the heritability, which is divided into two, under broad sense heritability, we measure our data under field condition, which is equals to computed by genetic advance, which is the most important factors in order to for selection purpose, which is equals to K times the genotypic phenotype variance times heritability. K is considered as a selection intensity for most crops runs from 2.06 as a constant. When I came to phenotypic and the genotypic correlation, This indication was for inherent of association between two traits. When we, as we see from this slide, when we consider two variables are the association between traits. It may be grain yield, and it may with the grain yield with a number of pod. It may be with a plant height in order to selection for a breeding purpose. When I came to the genotypic correlation, genotypic and the phenotypic correlation is most of them, it is correlated between the covariance of two traits with under square root of the phenotypic times the genotypic of it's a correlation between we select two traits. When I came to under genetic divergence is the most important for clustering and for variability, to show variability between, and diversity of the different genotypes under we select for breeding purposes. This formula is in order to select for which trait is more divergent and which trait is less divergency for breeding purpose. Principal component analysis. This principal component analysis to estimate a variable for most contributing of differential traits. When I came to under result and the discussion part, days to 50 emergency, days to flowering, grain filling period, days to maturity, plant height, number of pod per plant, harvest index, and seed yield was analyzed. As we see from this table, most of traits are significant difference, except the number of primary branch and the number of nodes per plant, which is non-significant. When we came to under range mean, heritability and the genotype from this table, most of traits under genotypic variance, which is the most under days to DF is which is equals to days to 50 flowering, which is to 7.29, which indicates that there is a good indication for selection under genotypic coefficient variance, which is median. Accordingly, now we can cluster. When we came to under heritability, most trait is which is their heritability is around 1918 for a qualitative trait. It is possible for qualitative trait. When we continue under quantitative trait, we can select for breeding purpose. Most of time, the ratio of heritability is 0 up to 1. Under when it is up to 75, it is possible to select for heritability mostly for a quantitative trait. This indicates that when heritability is greater than 50, it is possible for the second for indirect selection for breeding purpose of these genotypes. The highest yield was recorded from these genotypes, which were higher than that of grand mean, and the lowest yield was obtained from genotype this on the second sentence, which is indicated low yield. It indicates that it may be affected by environmental factor due to different causes. It may be agroecology of this genotype is not performed around the steady area. When we came to GCV value is greater than 20% for biological yield and seed yield. When I came to phenotypic coefficient variance, which is greater than 20%, it indicates that it is possible to select for breeding of next selection. This is maybe occur due to lack of adequate variability of this trait. It may be hinder breeding work for improvement of this trait. This finding confirms that the heritability allows more success in selection so that it obtained superior progeny was selected individual larger. So we can select indirect selection to obtain more gain, according to this author. This implicates that above trait was less influenced by environmental factor. It is good for selection. When heritability value is high with a genetic advance, it indicates that additive genetic action can expression for this trait. This finding was similar with these authors, which confirm it is positive for selecting next breeding purpose. Medium genetic advance as a percent of mean, which is between these ranges 10 to 20, it was recorded for days to flowering, days to maturity, plant age, number of pod per plant, and the number of seed weight, 1,100 seed weight. When it is heritability is low, it indicates that the percent of it is difficult to select. It causes pilotropic effects. Association trait between phenotypic and the genotypic variances. This table indicates that most trait is positively correlated for genotypic and the phenotypic correlation, which indicates that significant difference between most of genotypic and the phenotypic coefficient variances. As we see under plant age, most of phenotypic trait was positively significant when we compare to other traits. Under number of pod per plant also, it is significant differences between genotypic and the coefficient. This correlation at the above diagonal and the below diagonal indicates that it is possible to select those traits for most significant differences for the next generation. This result indicates that the seed yield had a positive significant correlation with the days to maturity, number of pod per plant, and the biological yield, or the dry matter at both genotypic and the phenotypic level. According to this author, this finding is similar with the genotypic correlation is higher than the phenotypic. This indicates that the genotypic factor is contribute. Genotypic factor is contributed. This is genetic for breeding value. Genotypic value is more contributed than that environmental factor for most traits. This result exhibits that the biologically also positive correlation with the number of pod per plant and the number of seed per pod and 100 seed weight at a phenotypic level. This finding is confirmed with the finding of these authors who reported the coefficient correlation with seed and the days to soybean for its maturity. As we see from this table, this is genetic distance analysis. Simply, we can observe this is 100 genotypes we cluster according to their average of traits. For all traits, we cluster 10 clusters for all 100 genotypes. This cluster distance indicates that the intra and intercluster distance. It represents to indicate the genetic diversity among all genotypes for wide crosses. And as we see from the above table, from this table, we can see the intercluster range from 0 to in second cluster, which is indicates that 0 means there is a little distance between them. It is difficult for breeding purposes. Genotype which belongs to the same cluster with a maximum intercluster, they indicate that more divergent and hybridization between genotypes in order to produce wide variability. We can observe from this the Main diagonal indicates that the largest cluster distance, which indicates that the interclusters. The main diagonal indicates that the intercluster, cluster within a clusters, and other clusters are between clusters. Principal component analysis. This result shows that 60, which contributes 61.6 total variation among genotype of soybean, which is the first principal component, which indicates that 23.6%, which is highly variable. In this present study, the differentiation of genotype between clusters is relatively higher contribution than the smaller one. We can observe this 100 genotype, which is clustered into 4 principal component analysis. Most trait under the first principal component is, which is for a number of primary branch per plant, number of pod, and grain yield, which is we high principal component analysis, high value of variation. This trait contributes that for implication of major contribute of trait. Most of trait are highly contributed under first principal component analysis. This present research indicates that with agreement of this most of authors for the performance of economically for important traits for the next breeding purpose. When I came to under conclusion and the recommendation, this result indicates that most of traits were the most important stated, which is positively and significant correlated. Generally, with a higher harvest index is greater than 45% were recorded for all genotypes. For most of trait, also heritability is less than 40%, which is possible to indicate for next breeding purpose, for selection purpose, in order to for hybridization. This indicates that improvement of seed yield simultaneously through indirect selection for most of traits. When I came to the last for us a recommendation, this experiment was done in a single location, which is difficult to select under a single location. It must be into multi-location with divergent hybridization between genotype, which is within interclusters in order to produce a wide variability for the production of a soybean genotypes around the location. Lastly, I wanted to say thank you for hearing and the authors also thanks to Wollega University and the Jimma Agricultural Research Center for supporting material during this experiment. Lastly, I wanted to say thank you, Sarah Levin, for coordinating this webinar conferences. Thank you so much.